And now for the latest European research into bird flu in this edition of Futuris. Out in the fields again. Every winter, Dutch ornithologists take a close look at the wild Siberian geese wintering here. They capture, analyse and then set them free in order to monitor for any presence of the H5N1 virus. And indeed, the virus is back in Europe. Researchers are on guard, identifying how it spreads and trying to understand its effects. They don't need to go far to get some answers, as nearby is a major research centre, one of the most secret in Europe. It's a bastion for animal research. The moat prevents live animals from coming anywhere near the laboratories. Cattle are not allowed within a 5-kilometre radius, and the centre has a special right to kill stray and wild animals coming into its premises. Only small, specially disinfected cameras are allowed to film. Every week, the centre receives dozens of live animal samples, but also whole dead animals found by biologists and volunteers. Researchers work inside under tight security, hoping to increase their knowledge about some of the world's most contagious animal viruses, including the one that causes foot and mouth and, of course, bird flu. The culprit for the flu virus, H5N1, was first identified in Europe in 1959, but the strain was very different from its current highly pathogenic strain, capable of killing 80 to 100% of the birds it infects. Researchers now know that the virus has sharp points, which are proteins. In orange, protein H, hemagglutinin. That helps the virus stick to the cells. And in blue, protein N, neuraminidase. That facilitates the release of viruses from infected cells.
All different subtypes can be very lethal. For instance, take the virus which caused the pandemic in 1918. In this pandemic, between 50 and 100 million people died. That was an H1N1. In the Dutch outbreak in 2003, which was caused by an H7N7, one veterinarian died because he was infected with this virus. And now we have an H5N1 virus, where about 200 people were affected and half of them died. So why these viruses are so dangerous for humans, we don't know. That's currently investigated by many research groups, and several characteristics came up. One of the proteins, internal proteins, seems to have a certain characteristics which makes it so dangerous for humans, and also the hemagglutinin will probably be very important for whether the virus is able to infect humans.
Guus Koch is an expert on bird flu. Indeed, he's been researching poultry diseases for 23 years. His team is currently participating in 6 different European projects, from developing vaccines to setting up early warning systems. Shared objectives, shared techniques. Most European research involves the inoculation of chicken embryos with the N5H1 virus. Researchers can then test how virulent it is and learn more about the pathogenic profiles of different strains. European researchers are still unsure about how and when the current strains may mutate to produce a pandemic virus in humans.
It's very difficult for us to know when the virus will change into a virus which will cause a pandemic. The virus then should transmit easily from humans to humans. And currently that doesn't happen. We don't know why that is. One possibility is that the virus currently is only replicating deep in the respiratory tract. And because of that, we don't produce saliva, and therefore we don't transmit it to other humans. If the virus would change in such a way that it replicates in the upper respiratory tract, so in the throat and in the nose, then maybe this could change.
More answers are also being sought near the city of Greifswald in northern Germany. This building hosts the Friedrich Loeffler Institute, another major European research centre into animal health. 8 different research departments and numerous laboratories are trying to unveil the mysteries behind animal diseases like Creutzfeldt-Jakob in cattle, classical swine fever, blue tongue in domestic cattle and goat pox, and of course, bird flu. Researchers here think that one of the best ways to isolate H5N1 virus and to prevent its spread to humans is to develop new and more powerful and more effective vaccines for animals. These chickens are being used to evaluate existing injectable but inactivated avian flu vaccines. But researchers here are looking further. They hope to design a whole new generation of the so-called recombinant vaccines.
We must be allowed to develop more recombinant vaccines here at the Institute of Molecular Biology. We aim to improve them and to make them more easily available, and we also want to improve diagnostic methods, enabling decisions over doubtful infections to be made more quickly.
Molecular researchers work to develop recombinant vaccines by mixing genes from the proteins of a virulent virus with the DNA of a non-virulent virus, which is then allowed to reproduce itself. Better understanding of the way H5N1 virus affects birds, cats and other animal species will also help to improve diagnosis and find better ways to distribute the vaccines.
For the
moment, we think that the distribution of vaccines via drinking water or sprays could be a good idea. The problem is that we might have to vaccinate large numbers of birds, and not just once. Doing it on an individual basis is not efficient, and we might miss some. However, by adding a serum to water or into a spray, one can quickly cover a whole population.
Recombinant vaccines may also prove useful in offering longer periods of effective immune protection. But European researchers agree there must also be respect for the health and security of the wider animal and poultry environment. Otherwise, H5N1 is set to mutate even faster than nature intends.
Changing of the virus might occur, but that's only if the vaccination is not applied in a proper way. We know from the situation in Mexico, where they vaccinate now for 10 years, without applying biosecurity measures, that indeed the virus did change over 10 years of time into a virus which is not protected anymore by the vaccination.
A world away from the peace and quiet of Greifswald's harbour, fighting H5N1 is firmly at the centre of a huge European research effort, having received a budget worth almost €42 million over 15 years. Indeed, there are dozens of EU programmes aimed at improving preparedness, detection and control of avian flu.